And why are we using copolymers? Yes, we're huge fans. Copolymers are definitely way better than detergents. I'm always on team copolymer because with copolymers, we can work 100% detergent free. We can retain the native host lipid environment because we can directly solubilize from the native membrane. We can very well stabilize and preserve the function of all different kinds of membrane proteins. And now we can finally also perform assays on body temperature, which was very hard to impossible with detergents. Therefore, we have lower risk and faster timelines. This is for our industry partners especially interesting. And also first time we were able to work with wild type GPCRs without any modifications. So that's the main reason. And there is a lot of copolymers out there. So at the moment in our portfolio, we have 32 different copolymers. Those are the classical SMAs we know from Michael and Barry Bruce and all consorts. There's the DIPMAs, we have the ASTIs, we have the SMABZs from Gestel and Bert, we have the ultrasolute amphipoles from Manuela, and we also have our own backbone, the Cubipoles, which is our own invention basically from our chemistry department. And this is the first backbone where you have all kinds of, or what does Michael Erkelen's always saying? He says this is the only copolymer backbone which has all site modifications on the market combined in one backbone. Yes. So in total, it's 32 different copolymers from 5 backbones. And we have seen that there is not one single copolymer which works best for every membrane protein target. So therefore, I'm always saying screening is key. And this is a lot of work. In addition, if you later want to do SPR, for example, we can fuse a biotin moiety directly to the copolymer backbone. Then you don't have to modify your membrane protein anymore, and you can directly immobilize this on the chip. We can also put a fluorescein on there if you want something sparkly there, or we could also, in theory, put custom modifications on there. Yes. So far, this is basically the workflow we all know. You have to resuspend the cells, lyse the cells, clarify your lysate, then you pellet your membrane, you add the copolymer, and you incubate it often overnight at 4 degrees. Takes a whole night to solubilize. Then you clarify your solubilizate, you bind it to your affinity resin, you wash it very thoroughly, and then you elute it. So this takes around 20 hours and it's a lot of manual labor. And so we were asking ourselves how we can save this precious time, how we can be more efficient, and also how we can condense the screening work. Because if you think you have to do all of this with 32 different copolymers, you have to do everything by hand, pipette everything by hand, you need an ultracentrifuge with like a small rotor. So this is an investment, it takes time, and it takes a lot of effort. So there are the points where we can attack. So can we maybe skip the membrane pelleting step? Can we directly work with clarified lysate or with full cells? Can we shorten the copolymer addition, maybe by increasing the time or by changing our habits on how we solubilize? Do we even have to clarify after solubilization? Is this even necessary? And can we maybe attack here at the binding for the affinity resin to save some time? And so what we developed here is a semi-automated workflow using a Kingfisher Flex system. This is a 96-well based system. And what we do is we have our cell suspension, we lyse it via sonication, but you can use whatever you want. We clarify our lysate, and then we can directly use this clarified lysate into a 96-well plate. And what we have developed is a 96-well plate where we can directly lyophilize the different copolymers inside. It can be all 32, it can be less. So if you go with the full 32 different copolymers, you could screen 3 different targets in one go, basically. So this is already good. You don't have to pre-buffer your copolymer anymore or like manually pipette it is already in there. And then in addition to this, we have also developed a 96 well plate where the magnetic beads are already dehydrated inside. So this is very nice, very neatly. I'm going to show you later how this looks. They are storable. You can just ship them at room temperature. You can just store them in your lab, and then you rip off the plastic, you aliquot it, and you can directly use it. And this is then all optimized for the Kingfisher Flex system so that we can then end up with 96 different stabilized membrane proteins or one membrane protein 96 conditions or whatever you wish in a very, very short time. So this is actually how it looks. This is how the rehydrated or dehydrated magnetic beads look in the bottom of the Kingfisher well plate. It's very accurate. We tested it on multiple different targets, and you can get very neat results. And now you can directly buy them from us as pure HT plates with indigo inside. This is for his purification of streptavidin XT, but we can also do any other custom resin. And in our paper and our work, we usually use the Rho1D4 tag. It's an antibody-based tag, which works very nicely for membrane proteins. Okay, so after all the optimization, we ended up with a 1.5-hour protocol. So this is already a huge improvement to the 20-hour protocol. And as I said, we can purify directly from lysate or from clarified lysate. We have a rapid solubilization with the Kingfisher system, which actually only takes 5 minutes at room temperature. This is great, and we were able to reduce the binding time with a bit of tricking for the row to only 5 minutes. And this then gives us a super quick protocol where we basically then have 32 copolymers on 3 different targets and all of this in 90 minutes tops. So this is really, really cool. Okay, this is how it looks so that you also believe me that I'm telling the truth to you. So you see that after 5 minutes binding time, you already reach 100% binding capacity with the Rho1D4 resin. And here's a corresponding Fomassi, which also shows you it's super pure. Already after 1 minute binding, you get super pure results. And this is a membrane protein P2X4. I'm also going to show you later a few results on. So with the robot, you not only get very quick results, but you also get very beautiful, beautiful gels, beautiful protein. Okay, so now that we had set up this technique, we were wondering, is it only working for one protein or can it work on many different membrane protein targets? And we have used GPCRs, we have used claudines, we have used SLC transporters, CD proteins, an EGFR, and all of this, 13 different proteins in total. And then we challenged our system to see if it really works. And conveniently, we also tested all the 5 different backbones to see maybe is there a winner, is there one copolymer backbone which performs or outperforms all of the others, or is it still is there still a reason to screen everything? And interestingly, what we saw over these 13 targets, we used 15 different copolymers for each target, and I've analyzed all of the gel bands on all of the gels, and what you see here is the best performing copolymer for each specific target. So for example, here it was ultrasolute aging, here it was one ASTI, here on the Claudin 4 sulfocubipol performed best, here sulfodipma performed best, here glycodipma performed best. So you can see, you cannot say there's one copolymer which ruled them all, not even SMA 200 as the old gold standard, but there's a variety of copolymers which can work very well. But what we could see, if we analyzed all the gels and all the copolymer stabilized membrane proteins, we can see a trend. And we compared with SMA as the gold standard, and you can see that DIPMA and the BZ polymers are not significantly different in output, but the ASTIs, the ultrasolute amphipoles, and the cubipoles, they outperform the classical copolymer backbones in forms of output. So in quantity you can get from the same pellet. And to not only bias in a gel, because it's sometimes hard to quantify, we also use the technique to look at raw fluorescence at 330 nanometers. And this is buried tryptophans, which are very common in membrane proteins, which are neatly folded. And we use the nanotemper penta device for this, and it can scan at 330 and 350. And we correlated this, and you can see that the output is rather similar. Also here, we see that the ASTIs, the ultrasolute amphipoles, and the cubipoles, they perform in generally better than DIPMA, SMA, and BZ in kind of quantity. So we're going to take a look at these 6 targets so that you can see the full gels and appreciate all the work we have done. And this is then how the screen looks. So up here, you can see the corresponding protein band. You can see that in this case, at the quantification, DIPMAs performed very well. SMA didn't perform so well, and there was no significant difference between BZ, ASTI, ultrasolute, and cubipol for the gel analysis. Here it looks a little bit different. Here you can also clearly see on the gel that the next generation copolymers, they gave us a better output. This is also true for these two targets. You can clearly see on the gel the bands, they get way stronger here. Also on corresponding Western blots, you can see this. Then of course the analysis also shows us this, that there's definitely a difference in output. And then again here, you can also see that it was rather similar with a bit of an advantage here. Interestingly here, the ASTI 1150 didn't work at all, but 650 worked very good. So this is the size difference we are often talking about. A 6 kilodalton copolymer sometimes performs significantly better than an 11 kilodalton copolymer because it's a little bit more aggressive in solubilizing. And the Claudin 1 worked very well in general. But also here, the BZ was performing very good, and also ASTI and Ultrasolid were performing well. Okay, so now we know that there is a difference between classical copolymers and next generation copolymers, at least in the 13 different membrane protein portfolio we are tested. But it's not all about the quantity on the gel. You also have to have a working particle. You have to have membrane protein which is happy in the environment, which is active, which is correctly folded. And this is nothing a gel could ever show us. And therefore, we now concentrate on P2X4. This is the example I also showed you before. And we can see here on the gel, again, the next generation copolymers, they perform better than DIPMA or SMA, in this case also as the BZ ones. And if we now look at biophysics, I picked three examples, glycocubipol, ASTI 655, and SMA 200. And you can see in the hydrodynamic radius, they are rather close together. Here again, you can see that the cubipol is around 6, while the ASTI is around 8, and the SMA is in the middle with around 7.5 nanometer. But if we now look at the stability of the particle, and what you can see here is a temperature curve, which goes from 25 to 95 degrees. And then we can see here the first derivative between 330 and 350 nanometer. So it shows us if the complex is unfolding. And you can see that the SMA unfolds around 50 degrees, the ASTI unfolds around 55, and the cubipol is stable until 65 degrees. So even though they look rather similar on the gel, they look rather similar in size, there's a difference in stability of the whole particle, which is a very interesting feature. Okay, then we thought about what is all important for a stabilized membrane protein. So I want it to be thermostable. This is the TM. I want to have a rather small hydrodynamic radius. Because if the hydrodynamic radius is too big, you often have nanodiscs sticking together, or it's just very clumsy and it's not going to refold finely. It's not going to bind as well. It's not going to look good in cryo. We need a very low PDI, the polydispersity index, because we want monodisperse samples. We want samples which are not very turbid, because we want to have a clean sample. And of course, we also want to get as much out as possible. And so what I did here is I looked at the full data set of the 32 copolymers stabilizing this P2X4, and I ranked them in a ranking system with these percentages, whereas if you calculate all of the scores together, one is the highest. And you can see here, this is the TM of the nano-DSF experiment. So how stable the particle is, and this is the hydrodynamic size. But for the score, I looked at all of these factors. And you can see that the cubipoles here in green, they perform best. Then the ultrasolid amphipoles, they were right after. The BZs and the ASTIs, they were somehow here in the middle. And the DSMAs, unfortunately, in this experiment in the very bottom. And the DIPMAs, they were very big. There's not even all of them shown. One was 60 nanometer. This even fell out of this calculation. So this could be a tool for you to assess the protein quality, and especially if you want to do drug screening or if you want to do cryo after, which gives you the best fitting copolymer for your specific target. So we purified this in a little bit of a bigger scale, and we looked if the protein is correctly folded and if it's correctly glycosylated. Because P2S4 is heavily glycosylated, we did a PNGase assay where you can see here a clear shift. So we have a nicely glycosylated sample. P2X4 is around 50 kilodalton. It's a trimer. A natural trimer should be around 150. Also, this fits very well. And then we use two different techniques to assess ligand binding. Those are both small molecule ligands. So we have 150 kilodalton protein and something around 200 Dalton, 300 Dalton small molecules. So this is very, very hard to assess. And we used a GCI, either in classical SPR mode or in this wave rapid technique. And we used an MST assay with a spectral shift. So this is an in-solution assay, and this is an assay where the particle is immobilized on a gold chip. And we can see that with both techniques, we were able to get nice KDs, and those also fit in the classical literature values. So we can say our P2X4 is not only looking beautiful on the gel, very thermostable, but it's also active in two different assays with two different ligands. And then we thought, okay, this looks so good, maybe we can even solve a structure of this. So we went directly from the illusions of the robot on grids. And this was very lucky. It's not at all something which you manage with every target, but in this case we were. And with 4 different copolymers, we were able to get high resolution structures ranging from 3.5 to, I think, 2.9 angstrom. So this was really, really cool, really, really lucky. And you can here see the grids, how they are looking, and 2D classifications. And this is our 3D model. We even catched endogenous ATP. So it was binding ATP so tight, we didn't add it during the purification, but it retained its shape there in our structure. Yes. And with this, I want to thank the whole team. If you want to check out the paper, this is the title. It's on bioRxiv. And yes, I'm happy for any questions.
Thank you so much, Philipp. Fantastic work by you and the team. This is very exciting to see how quickly you can actually run through all of these polymers and the valuable data that you can get. So that's very exciting. And I see there's quite a few members in the community that's also excited about this. So we have some time for questions. Let's ask our first participant. Surah, if you want to unmute yourself and ask your question regarding the use of your particular target. Essentially, they're asking whether you've tried these copolymers on the SLC25 family.
Yes, we have used also the SLC family, and we've used all the copolymers on there. We actually have a list with more than 100 different full-length membrane proteins you can check out. On our website, there's also details. I don't know for the specific protein right at the moment, but SLCs are definitely in the portfolio, and they're not easy to work with.
Okay, so let's go have a look at the website for some more information on that. And then, Michael, would you like to ask your question?
Sure. Did you see any bound lipid head groups?
Yes, we did. But for this, exactly, I have to ask my colleague.
Okay, so it's in the paper on bioRxiv or no?
Yeah. Sorry again?
Do you have the lipid structures bound to the target in the bioRxiv paper that you've uploaded?
There's definitely some structures, but I don't know if those are exactly in there. But else, yeah, I can ask my colleague for this.
Okay, great, thanks.
Let's ask 1 or 2 more questions. I have a quick one. Practically speaking, I see that you, after you have your clarified lysate, how much work went into essentially optimizing that centrifugation step to get rid of anything that might interfere with your downstream processing? Did you have to try different centrifugation speeds to get to that 9,000 that you recorded?
Yes, we tried out a big range, but actually the classical 9000, which everybody's using in publications, is working very, very nicely. We just added 50 minutes on top because you often have this whitish blob when you pour over your lysate from the centrifugation tube. And centrifuging it for 45 minutes makes it significantly easier to just pour it over. So that was basically everything. But this clarified lysate, it works super fine. I would have never thought. I was always using membrane pellets. But yeah, it works fine.
Fantastic. Thank you. I think that can also overcome quite a few hurdles in the implementation if we can get rid of that long ultracentrifugation steps.
Yeah.
Thank you, Philipp. And then I think there's a last question over here from Anthony. Anthony, would you like to ask your question? If not,
Hi.
oh, there we
Sorry.
go. Perfect.
Nice talk, Philip. I was just wondering, is there a property of the copolymer that would determine the final size of the nanodiscs formed?
Yeah, so what we see in all of the targets we have purified is that there's no size difference between the different copolymer backbones once you have a purified sample, which you run over a sec. So you have a really polished sample. In my experience, or in our experience, it doesn't matter if it's an SMA, a DIPMA, a BZ, a ASTI, a Kubipol, whatever, or nitrosylate amphipol, to name them all. In the very beginning, after elution, once it's not super clean, you will definitely see differences. And if you just use lipids like a POPC or a DMPC, you're going to definitely have size differences.
Okay, thank you so much, Philip. There's 1 or 2 questions in the chat. If you can have a look at those during the next talk, that would be wonderful. Let's thank Philip and let's welcome on